like to thank Abby King, who's serving as the chair of Amy's committee, other committee members who are all here, Professor Ali Boehm, Professor Grant Miller, Professor Pavani Ram. So thanks to everyone for being here. I'm Jenna Davis. I'm a faculty member with a joint appointment between Civil and Environmental Engineering and the Woods Institute for the Environment. And it's my great pleasure to introduce Amy Pickering at her defense today. I'm one of her two lead advisors for her work in E-IPER. And many of you know a lot about Amy, but just to recap, she has an illustrious career, starting with an undergraduate degree from Cornell, Environmental Engineering and Science, a master's in Environmental Engineering from Berkeley, and then will be completing her PhD in E-IPER here at Stanford. We're very lucky to have her. Her work in E-IPER really embodies, I think, what the program is about, drawing on multiple theory bases, conducting rigorous research of the highest quality that both has immediate applications for policy and decision making, but also is extremely fruitful in different disciplinary intellectual communities, sort of identifying important intellectual gaps, puzzles, debates that need to get worked on. So her work is tremendous, and we've asked a lot of her in the work that she's done. Many of you in this room have worked with Amy in the field, and so you know that we've asked her to take samples in 100-degree heat. We've asked her to get up and close and personal with toilets a lot, work in the lab, motivate and supervise dozens of enumerators that she's trained in different countries around the world, and also working with other students as research assistants here at Stanford. So she's done a tremendous amount in a wide diversity of environments, and this has led to an incredibly productive time here at Stanford. She currently has 5 peer-reviewed papers stemming from her dissertation work, 4 of which she's first author on, and several more in the works. Incredibly focused and productive person, and we're all sort of in awe of her about that. But I should also say that she's goofy and fun, and really helps keep us focused on the fact that there is life and there is work, and they are different. And so she's made an awful lot of friends here at Stanford and beyond, and I know I'm not at all surprised to see how many people are here in the audience today to support her, as she has befriended and supported so many other people. Really important things to know about Amy: she likes ice cream, so anybody who's interested in congratulating her, take her out for ice cream along the way. And the other thing is that I think everyone in this room, or almost everyone in this room, could probably say that your hands are a little cleaner for having known Amy. So congratulations on all the hard work, and we're looking forward to hearing more about it.
So just to set the stage, Amy's going to talk for about 40 minutes. We'll then open it up for Q&A from the audience. And then we will retire upstairs to do the hard grilling that goes into these documents.
Okay, thanks Jenna for that wonderful introduction, and thanks to everyone for coming here today to listen to me talk about my dissertation on water access, hand hygiene, and child health in Sub-Saharan Africa. So I want to start off by just talking a little bit about what motivates my research, and that is global child mortality. So what we're looking at here is the number of child deaths that happened in 2009 as reported by UNICEF. And we can see that over 8 million children died in the year 2009 before they reached their 5th birthday. And most of these deaths are occurring in low-income countries. You can see here that almost 4 million children died in Sub-Saharan Africa in 2009. So why are all these children dying? So this is showing the causes of child deaths, under child deaths from age 1 month to 59 months. And what we can see is that the 2 leading causes of child mortality are diarrhea and pneumonia, shown here in blue and yellow at 24%. And these are really preventable diseases. So if we take into account the fact that most child deaths are occurring in Sub-Saharan Africa and that they're being caused by diarrhea, we get sort of an interesting picture of the world. So I call this the world according to diarrhea. And what's this showing here is countries in proportion to the number of child deaths that occurred there in 2002. And you can see that Sub-Saharan Africa is quite large. So how is all this diarrhea being transmitted? Let's look at a pretty famous diagram here that's showing how fecal contamination transmits diarrheal pathogens. So we see how when feces are excreted from an infected individual, they can travel along a number of different pathways, including through water, on hands, in food, on flies, and through fields. And so for example, in places where there's not good access to sanitation, feces can get into drinking water supplies or can get into food that's then consumed. And although we know that all of these pathways exist, what we don't know a lot about is which of these exposure pathways are most important and which really cause the most burden of diarrheal disease. And I also want to highlight that hand contamination has really been understudied in low-income countries. There's been a lot of focus on water quality and how fecal contamination and diarrheal illness is transmitted through water, but less so on hands. So the good news is that there are a number of interventions that have been found to reduce diarrheal illness in low-income countries. First is sanitation, so this means building improved toilets or constructing drainage systems or sewers, and this is sort of acts as a primary barrier to stop fecal contamination from getting into these other pathways that can transmit illness. Also, water treatment, either at the source or within households, can disinfect water and prevent ingestions of pathogens. And hand washing with soap and water has also been found to reduce diarrhea as well. So several meta-analyses have been conducted to try to understand what sort of average effect that we could expect in diarrheal disease reduction by these different types of interventions. And as you can see, all of these interventions can reduce incidence of diarrhea up to 45%. But what I want to point out here is there are not very many studies in these meta-analyses in the categories of water supply and sanitation. And that may be one reason why we see this discrepancy in water supply. It also might be because the definition of improved water supply is a little bit fuzzy. It could mean that a source is improved, like upgrading a spring to a protected spring, or a shallow well to a protected shallow well, or it could mean building a water source closer to a household. So let's look a little bit closer at access to water supply in the world. So this is a map showing the proportion of people that have access to an improved water source as defined by what's called the Joint Monitoring Program, which is an organization that was developed by the UN and the WHO to sort of track progress of the world towards Millennium Development Goal number 5, which is aim is to have the number of people without access to safe water and sanitation by 2015. So you can see in the red are countries where the proportion of people that have access to a safe water supply is less than 50%. And what I mean by improved water source is a source that's within 1 kilometer of a household and that's protected from contamination, like a deep bore well or a protected spring or piped water. But even though people have access to an improved water source, they still have to, in most cases, leave their household to go and fetch that water and bring it back. And in fact, over 3 billion people in the world are using shared water sources. And it's estimated that about 40 billion hours are spent each year carrying water in Sub-Saharan Africa. And we know that women and children are the main water haulers. What we don't know is what impact this has on health. So recently, a meta-analysis was done to look at the relationship between distance to water source and incidence of diarrheal illness. And although this meta-analysis did find a positive association between further distances and diarrheal illness, they did not have enough data to develop a quantitative relationship. And the fact of the matter is that most of the literature that's been conducted most of the research that's been conducted to date has really dealt with water quality interventions and types of water sources, and not distance and time that it takes households to fetch water. So now I want to walk through a conceptual model that I hypothesized that really has guided a lot of my dissertation work. So let's start out with water access. And so by this we mean distance to water source from a household to their water source and the time it takes household members to fetch water. And these things in turn likely affect the quantity of water that households collect, as well as how much time they're spending out of their daily time budget collecting water. And these two things affect a variety of behaviors. So the behaviors that we're most interested here are hygiene. So it's been found in several cross-sectional studies that the amount of water that a household has available is associated with how much water they use for hygiene purposes, like hand washing with soap and water or bathing. And in fact, households with piped water in East Africa have found to use twice the volume of water for hygiene purposes than households without access to piped water. Another pathway of interest is that households that are fetching water and storing it in their home are probably storing water for longer periods of time than households that have water closer to them. So this is a problem because stored water can become recontaminated. A lot of times water is stored in buckets that are open, where household members are having to use dip cups to extract water and so forth. Something that might not be as intuitive is the fact that time spent fetching water is not being used for things like childcare, seeking access to health care, schooling, and even income-generating activities. Like, for example, households that have more time in water might be able to grow crops and sell them in the market. And an example of how time might be important is that in a lot of low-income countries, healthcare seeking behaviors, they don't cost a lot of money, but they cost a lot of time. So for example, there could be a free health clinic several hours away that a mom needs to walk to with her child to get that free health care. So as I've mentioned, a lot of these behaviors affect hand cleanliness and drinking water quality. And the way that I'm going to be measuring these things in my research is looking at levels of fecal contamination on hands and in drinking water, and looking for the presence of diarrheal pathogens. And finally, the outcome that we're ultimately interested in is child health. And so today I'm going to be talking about diarrheal illness, but as well as respiratory illness, because hand hygiene behavior has also been found to be associated with respiratory illness, which is a precursor to pneumonia, which is, as we know, a leading cause of child mortality. I'm also going to be talking about child anthropometrics, specifically height and weight of children. And so these measures are being increasingly used in my field and other fields because they're really nice objective indicators of child health that don't rely on self-reported data. And it's also been found that children that are underweight and that are stunted are more likely to get ill, and they're also it also associated with cognitive function and development in general. And we'll also be looking at childhood mortality. So now I want to put the research that I'm presenting today in the context of this conceptual model. So the first part of research that I'm going to be presenting looks at the relationship between water access as measured by one-way walk time to source and child health in Sub-Saharan Africa. And we're not going to be concerned so much with these intermediate causal pathways, but we're just going to try to understand the relationship between these two things. The second part of research that I'm going to be presenting focuses on the right side of this conceptual model, looking at how fecal contamination is transmitted through hands and through water among households accessing non-networked water supplies. And I'm going to be focusing particularly on the role of hands. And then finally, I'll present an observational study that looks at how hands become contaminated with fecal contamination. And both of these field studies are conducted in Tanzania. So let's get started with the first part of research. So here I'm looking at the quantitative relationship between walk time to water source and child health in Sub-Saharan Africa. And to do this, I pulled data from what's called the demographic and health surveys, which are household surveys, nationally representative household survey data sets that's a program funded by the USAID to understand population health and fertility in low-income countries. And so in this data set, we have one-way walk time to water source that households have to walk to fetch water, the type of water source that households are using. We also have a number of child health indicators that we're interested in, including 2-week prevalence of diarrhea, cough, and fever. In addition, these data sets include measurements of height and weight of children. And the way that I'm going to be presenting these measurements are through z-scores. So z-scores are commonly used by the nutritional community to understand how many standard deviations above or below what a child should be if it's healthy. So if they have a z-score of 0, then they're healthy. Also, these data sets include GPS points of clusters of households, which turns out to be really useful. So, as a lot of economists and epidemiologists will tell you, if you have a cross-sectional data set, like I just described, and you want to understand causal inference, you run into some problems, because it could be that there are confounding or unmeasured variables that are associated with your independent variable of interest, such as water access, and your dependent variable of interest, such as health. So, for example, households that are closer to their water source might also be wealthier, which also could affect health. So, to address this problem, I use an econometric technique called an instrumental variable approach. And the idea here is to find a variable that's randomly distributed or can be considered randomly distributed and not correlated to confounders that you're worried about. So in this case, I selected freshwater availability. And so I used a 2-stage regression approach. The first regression regresses time on freshwater availability. And then I use those predicted values of time explained by the variation in freshwater availability and substitute into a second-stage equation where health is the dependent variable of interest. So there are 2 important assumptions for using this approach. The first is that freshwater availability is strongly correlated with walk time. And I'll address that in a moment. The second is that freshwater availability only affects health through the pathway of interest, which in this case is water access. And so with this analysis, we might be concerned that freshwater availability affects type of water source, which also could affect health because, for example, in places where there's a lot of fresh water available, households might be accessing surface water sources, which might have lower microbial quality than households accessing like deep bore wells or piped water. So I also control in this analysis for type of water source as a proxy for water quality. So a little bit about the freshwater availability data set that I used. It's generated by a water balance model. It includes variables like temperature and precipitation and land cover to determine the amount of fresh water available per grid. And the grid size in this data set is at the 0.5 degree level. So I took this freshwater availability data set and I overlaid my household survey data using the georeference points of clusters of households. So each red dot in this map is a cluster of households. And I end up with a nice data set that includes all of the child health outcomes interested in freshwater availability, water access, and I end up having data from 26 countries in Sub-Saharan Africa with about 200,000 children under 5 in the data set. So a little bit about the households in this data set. About a quarter of them are accessing piped water supplies. Only 7% have access to a flush toilet. About 35% don't have access to any sort of toilet facility at all. Diarrhea is pretty high at 17%, children under 5. About 22% of children are underweight. And so the way the WHO classifies underweight is if a child has a z-score below -2. About 41% of the children are stunted. And the same classification is used here. If the child has a score below -2, they're considered stunted. And mortality in the data set is at about 10%, under 5 mortality. So the mean one-way walk time in this data set to a drinking water source is 23 minutes. About 18% of the households in the data set have their own water source, so I substitute 0 minutes in for walk time for these households. And I do find that walk time to water source is negatively associated with freshwater runoff, so we find a strong first stage here. So now I'm going to show you the results from my instrumental variable models. So what we're looking at here is how a change in 15 minutes in one-way walk time to water source affects these various health outcomes. And what we find is that with a decrease of 15 minutes in one-way walk time, diarrhea falls by 7 percentage points. Fever falls by about 5 percentage points. Cough falls by about 4 percentage points. And we find that our child anthropometric scores are all increased. So children are less likely to have growth faltering if they're closer to their water source. We also find an effect on childhood mortality. So this change, the same change in one-way walk time to water source is associated with 1.4 percentage points lower child mortality. So if we think about those baseline levels of illness and mortality that we had in our data set, we can look at the relative reductions that this same change in walk time to water source is associated with. And we find that diarrhea, the relative reduction is 41%. And this is actually really comparable to those results that I was showing you earlier, showing you the average reduction you can get from water quality improvements and from hand hygiene promotion. I just want to mention that all of these models control for water source type, like I mentioned, whether the household's in an urban or a rural area, child age and gender to increase the precision of my results. So this is a figure showing you the relationship between walk time to water source and the predicted probability from my modeling results. And what I want to point out here is the red line is diarrhea. And we see that the marginal effect, or the slope of the line, is much higher. There's a much higher effect on diarrhea than for cough and fever. And this makes a lot of sense if we think about the causal pathways that we were talking about earlier by which water access could affect health, including that stored water could get recontaminated, that households might be less likely to be practicing hand washing, and so forth. So this is a figure showing the relationship between walk time to water source and the predicted Z-score for children. And you can see that as walk time to water source increases, then predicted Z-scores are falling. And again, I want to point out the slopes of the lines. So weight for age has the largest slope. And this makes sense because weight for age is more closely associated with incidence of diarrhea than height for age and weight for height. So some conclusions from this first part. We found that walk time to water source is associated with child morbidity and mortality in Sub-Saharan Africa. And it sort of suggests that walk time to water source should be given more attention as an indicator of water access. Also, current water supply investments in Sub-Saharan Africa are largely taking the form of improved shared water sources. So this is a figure showing how the percentage of people with access to piped water, the blue shaded region of the graph, has really remained the same in Sub-Saharan Africa from 1990 to 2008. Whereas in the rest of the world, it's been steadily increasing. And so these findings sort of suggest that these infrastructure improvements that are being built in Sub-Saharan Africa are likely not to have the health impacts that probably policymakers and governments are hoping for. Another interesting implication of this research that I want to touch on, and that is that there's been a lot of recent research lately coming out talking about how climate change and growing urbanization is going to lead to water scarcity. And so this is a map showing what's called the human water security threat. And as you can see, in places like Sub-Saharan Africa and Asia, where countries don't have the resources and the money to mitigate this water security threat by building infrastructure, we see water scarcity. And so these findings suggest that these changes in water scarcity or water access are going to have significant impacts on child health. So now I want to move on to part 2, looking at how fecal contamination is being transmitted through hands and through water among households in Tanzania accessing non-networked water sources. And the first objective of this study was to look at what household characteristics are associated with high levels of hand contamination among mothers and children. Second, I wanted to look at if there was an association between the level of contamination on hands and the level of contamination in stored water within households. And then thirdly, I wanted to look at if hand contamination or water access, or not water access, water contamination was associated with child health. So we did this study in Dar es Salaam, Tanzania. We worked in 3 communities that had actually recently received from the World Bank improved shared water sources in the form of deep bore wells. And what we did is we used a weighted sampling approach to select 334 households with the selection criteria that each household have at least 1 child under 5. And we collected stored drinking water samples from these households in sterile bags, and also hand rinse samples from children under 5 and from mothers. And the way that we sampled hands was we had the mother or the child place both hands into a bag of sterile water, and we would rinse the hand, and the bacteria would come off into the water. We also sampled drinking water sources in these communities, and these included bore wells, fenders, and the pipe network. So we took these water and hand samples back to our field lab in Tanzania, where we processed them by membrane filtration. And the way membrane filtration works is that you pass water through a filter, and then you take that filter and you put it on media, and you culture bacteria in an incubator. So this is an example of a plate from this process that's growing E. coli. And the blue colonnades indicate that one E. coli was present in the sample. So you can count them up to get the total number of bacteria that was present in a sample. We call these colony-forming units, and I abbreviate these as CFU later on in the presentation. The 2 fecal indicator bacteria that we were looking at were E. coli and Enterococci. And these are commonly used to indicate the presence of fecal contamination in water. And I just want to mention that these fecal indicator bacteria, they indicate the presence of feces, but they don't necessarily mean that actual infectious pathogens are present. So this is a figure showing you the level of fecal contamination that we found in stored water, in source water, and on hands. And on the y-axis, we have log-transformed E. coli per 100 mils for water samples, per 2 hands for hand samples. And what we find is that there are much higher levels of fecal contamination in stored water samples within households than from the sources that they were collecting water from. And we also find really high levels of fecal contamination on the hands of mothers and children. So this is showing you the average contamination level on hands with 3 log. For those not familiar with log units, that's over 1,000 bacteria per 2 hands. And to put the water quality results in perspective, the international guidelines for drinking water are 0 E. coli per 100 mils of water. So these are the model results of a model that I constructed to look at how household characteristics are associated with hand contamination. And it's a multivariate regression model, and the dependent variable is log E. coli and log Enterococci. And what we find is that mother's education is associated with lower levels of bacteria on hands. Perhaps this is because mothers with more education are more knowledgeable about germ theory, and so they might practice better hygiene. We also find that an improved toilet, and so by improved toilet I mean a toilet that has a reinforced pit or is connected to a septic tank, is associated with lower contamination on mother's hands. Also find that an infant in the household is associated with higher levels of contamination. And we also find that for Enterococci, if a mother reported being dissatisfied with the quantity of water that she had available for hygiene purposes, then she had higher levels of contamination on her hands. So to investigate the association between water contamination, stored water, drinking water contamination in a household, and hand contamination, I looked at the correlation between these two things. And I found that there was a significant positive correlation between the level of bacteria in stored drinking water and the level of bacteria on hands. And so this sort of provides support for the hypothesis that dirty hands might be contaminating stored water. And to further support this hypothesis, I found that water extracted by hand dipping, like a cup or a mug, into a stored water container was more contaminated than water containers where the water is extracted by pouring or by a spigot. Also, when I modeled bacteria in water quality as a function of a bunch of different household water management practices, like reported water treatment, whether or not the water source was covered, where that water source came from, how long that water was stored, found that bacteria on hands was the only significant predictor variable in the model. So now let's look at how water quality and hand contamination was associated with child health among these households. So to do this, constructed binary logistic regression models with whether or not a household had reported a child under 5 having diarrhea or respiratory illness in the past 48 hours. And so these models control for education, whether the household was a single home or a multifamily home, weekly expenses is a proxy for household wealth, and whether or not the household had an improved toilet or not. And what we find is that levels of bacteria in water are not associated with health, but we find that bacteria levels on hands are associated with health. And in fact, a 1 log increase in enterococci on hands is associated with the odds of a household reporting diarrheal illness increasing by 10-fold, and the odds of a household reporting respiratory illness increasing by 2-fold. Interestingly, we see this opposite association with E. coli on hands, suggesting that higher levels of E. coli on hands are associated with lower levels of respiratory illness. And don't really know why that's the case. It could be because E. coli is not similar to the types of pathogens that cause respiratory illness. It also could be because people with runny noses and coughs might be more likely to be rinsing their hands more often. So some conclusions from this second part. I found that hands seemed like an important transmission pathway for diarrhea and respiratory illness, and maybe more important than water quality among these households in the study. And so this suggests that successful hand hygiene interventions could improve stored water quality within households and also improve health. So now I want to move on to the third part of research that I want to present today. And so this study was really designed to understand how are hands becoming contaminated with fecal contamination. We're finding these really high levels of bacteria on mothers' hands. How are they getting there? So the main research question is, how do typical household activities that a mom does during her day affect levels of hand contamination? And then we were also looking at if we could actually find diarrheal pathogens present on hands. Because like I said before, fecal contamination doesn't necessarily mean that there are pathogens. So we wanted to look for actual pathogens to sort of gain some insight on what are the actual health risks of high hand contamination. And before I move on, I just want to show a short video. So this is a mother in Tanzania. She's wearing a head camera. She's just touched some charcoal. And now she's sort of pausing before she goes to where she's got some water from where she was doing some laundry, and she rinses off her hands. And I think this does a nice job of sort of demonstrating the conditions under which these moms are dealing with all the things that they have to do during a typical day. You can see the dirt floor. There's no access to piped water. So the research design that I used for this study involved structured observation paired with repeated hand rinse sampling. And we had 2 cohorts. So we enrolled 119 households with the selection criteria that each household had to have at least 1 child under 5. And we had 2 cohorts, an activity cohort and an observation cohort. I'm going to explain to you now what those are. So in the activity cohort, we had about 100 moms. And what we did here is we arrived at the home and we took a baseline hand rinse sample. Then we asked her to wash her hands with soap and water, and then we took a second hand rinse sample to document that reduction in bacteria. Then we asked her to perform a specific activity. And these included sweeping, food preparation, dishwashing, bathing, cleaning the toilet, defecation, urination, cleaning up a child after they had defecated. We also had a control group where our mom sat with her hands on a clean paper towel in her lap. After she did this activity, we took a final hand rinse sample to look at how bacteria had changed on the hands. In the observation cohort, we observed moms for 8 hours. And what we did is we took hand rinse samples every 2 hours. And enumerators sat and recorded everything that the mom did and everything she touched, and also when she washed her hands and if she used soap or not. And the idea here was to understand what activities are contributing the most to fecal contamination or reductions in fecal contamination on the hands. So we processed these hand rinse samples in the same way that I described in the previous study to look for the 2 fecal indicator bacteria, E. coli and Enterococci. But we also looked for 2 diarrheal pathogens. And so we looked for enterovirus and pathogenic E. coli. And these 2 pathogens have been found in the feces of children in Tanzania that have gone to the hospital with diarrhea. So for enterovirus, we looked at a subset of baseline hand rinse samples. The way we did this is we filtered a portion of these samples, and then we brought those filters back to Stanford, extracted the RNA, and used RT-PCR to screen for enterovirus. For pathogenic E. coli, we grew up E. coli on filters, and then we brought it back to Stanford and extracted the DNA and used conventional and multiplex PCR to look for 3 specific strains of pathogenic E. coli. So a little bit about the households in this study. About 10% have water source on their premises. They are spending a significant amount of time fetching water. Almost all the households have access to a toilet, but a lot of them share this toilet with other families, and most of these toilets are unimproved toilets, not connected to a network sanitation system. So I want to show a few example results from the activity cohort. So what I'm going to be showing to you here are plates where we cultured E. coli bacteria. And we did 2 different filtration volumes to improve our detection limit. So the inside column is going to have less bacteria than the outside column. So this is a baseline sample. This is after hand washing with soap and water. You can see how bacteria levels decreased. And then after the activity, on the left, washing dishes, defecation on the right, we see those bacteria levels spike back up again. So let's look at these results in a little bit more detail. This is a figure showing the specific activities on the x-axis, ordered from lowest increase to highest increase. And on the y-axis is the geometric mean increase of bacteria per 2 hands. And I want to point out first the control group. So this is Enterococci, and in the sitting group, we found that levels of Enterococci remained about the same. You can see that arrow bar crossing the x-axis. And I'm not showing the E. coli results here, but E. coli actually decreased for this group, perhaps because it died off. So for all these other activities, we see significant increases in bacteria on the hands. Even activities that you wouldn't normally think of as fecal contamination events, like sweeping, doing dishes. And I want to highlight the highest level of increase that we observed was for the group where the mothers prepared food. I'm going to come back to this. Now moving on to the observational cohort. So here, remember we were taking hand rinse samples every 2 hours. So you can see on the right here an example 5 plates from one mother's hands. And you can see that there was a lot of variability throughout the day. So to analyze this data, what I did is I aggregated the data by 2-hour increments and matched up the observational data with the hand rinse sample that was taken at the conclusion of each 2-hour interval. And I used generalized estimating equations to account for correlation within the same mom. And so these used hand bacteria counts as dependent variable, and specific activities were included as binary independent variables. And I also included the time that had elapsed since the last hand washing with soap and water. So this table is displaying the results from that model. with log enterococcus per 2 hands is a dependent variable. And what we find is that a mother leaving her household, a mother handling food, bathing, and the time since last hand washing with soap are all significant variables in the model. The way we can interpret these results are if the mother had prepared food in that previous 2 hours, she was likely to have, on average, 0.8 log more enterococcus on her hands than a mother who had not prepared food in that previous 2 hours. And so this matches up well with the activity data that we had just looked at, where we saw that moms, when they prepared food, there were high levels of bacteria on her hands after that, as compared to hand washing with soap and water. So in terms of pathogens, we did find them on hands. For enterovirus, we found about 9% of the samples that we screened were positive for enterovirus. And for pathogenic E. coli, we found about a third of the samples that we screened for pathogenic E. coli had at least one strain of pathogenic E. coli in the sample. So some conclusions from this third part. We found that food handling increases fecal contamination on hands. And this could be because that in a lot of low-income countries, produce is grown with sewage. And so that might be contaminating food. It also could be because food is sold in really unhygienic conditions in markets. I don't know if anyone's ever been to a market in Africa. It can be pretty disgusting. We also found that bacteria is reduced by hand washing with soap and water, but it's also affected by a number of other activities, making bacteria really highly variable on hands. And finally, we found pathogens on hands, and this suggests that there is some measure of health risks associated with hand contamination. So to summarize everything that I presented today, I want to go back to my conceptual model. So we found that walk time to water source was indeed associated with child health, suggesting that water access should be given much more attention as an indicator of water access. Currently, the UN considers a source within 1 kilometer of a household to be improved, or as a measure of good access. And we found that even cutting that, and most often, 1 kilometer is considered to be a 30-minute walk. So we found cutting that in half could lead to a reduction of 41% in diarrhea and 14% in child mortality, which are pretty significant reductions. Second, we found that hands play a pretty important role in transmitting fecal contamination and illness within households accessing non-networked sources. So if we go back to our diagram of fecal-oral transmission pathways, we can add an arrow now from hands to stored water quality that wasn't there before. And third, we found that mom's hands are contaminated by a variety of activities that she does in her typical day. And this suggests probably 2 things. One is that environmental contamination in these communities is probably pretty high. And second, there's been a lot of attention lately on trying to identify good indicators of hand washing behavior in the development community, because there are a lot of money going into trying to promote hand washing as a low-cost way to reduce diarrheal illness. And people are looking for ways to measure the outcomes of these promotion programs. And a lot of people have suggested that fecal indicator bacteria on hands could be a good objective indicator. But we find here that it's pretty highly variable, and some caution should be used when it's used as an indicator of hand washing behavior. So now I just want to touch on some potential next steps from this work. So we spent a lot of time in the right half of my conceptual model. I think it would be really valuable to better understand how water access affects the quantity of water that households are collecting, and how much time they're spending on water fetching. So it would be really nice to know if you could save households time fetching water, what will they do with that time? Will they do better childcare? Will schooling increase? What will happen? Also, if households have more water available, what will they do with that water? Will they grow more crops? Will they do income activities like brick making, et cetera? This could really give us an idea of what other impacts besides health improving water access might have. And finally, I want to conclude on a sort of a positive note. The last 2 chapters of my dissertation deal with looking at waterless hand sanitizer as a potential solution to improving hand hygiene in water-constrained environments. And so the work that I've done so far has shown that the efficacy of waterless hand sanitizer can be as good as hand washing with soap and water in field environments like in Tanzania. And so I think that the next steps to this work are to look at how promotion of waterless hand sanitizer, what sort of health and behavioral impacts it can have. With that, I want to acknowledge my funding sources, and I'm going to take questions now, and I'm going to do some acknowledgments after the questions. Jesse?
I wonder if you compared the hand samples that you did in Africa with samples that you took in a place where people might have cleaner hands, because you were saying 9% had one type of virus. Is that typical across the world, or is that really bad?
So that's a great question. And actually, I wanted to mention this when I was presenting the results, that we actually measured the hands of children in daycare centers in East Palo Alto, and we found that 95% of their hands were completely clean of E. coli, no E. coli on the hands. In terms of looking at actual pathogens, there really hasn't been very much work done at all to look at actual pathogens on hands. There's been some work done in hospitals and healthcare facilities, but almost no work done like among people like you and me. Yeah.
So did you do any samples where you wash somebody's hands and then immediately wash them again to see if you actually removed everything?
Yeah, that's a really good question, and we did do that. So I did a sort of a small study where we did sequential hand rinse sampling to see how much bacteria we'd get off on that second hand rinse sample, and we were getting off a lot of bacteria. And we found that I can't remember the actual numbers, but maybe about 50%. You know, it was the levels of bacteria were changing, being reduced by about 50% each time we took a hand rinse sample.
So how did you deal with soliciting people to be part of the study when you were asking them if they do it? Did they know what the study was about, and how did you deal with like the fact that you just being there might influence what they did on a daily basis?
Yeah, so that's a great question. And what I didn't actually describe is that all of this field work was done by local enumerators. I was never at the households because a white girl always biases the results. So I also spent multiple weeks with my enumerators training them on how to approach households and how to explain the risks and the benefits of the study and make sure that they could totally understand how much time it was going to take and what was going to be done in a hopefully a culturally appropriate way. In the back, yeah?
Hi. Is there ever any intervention of things like potassium permanganate? I remember many years ago in Hong Kong when I was doing research, there was at various stages you have to vegetables. How does intervention of chemicals or bacteria antibacterial compounds and work here?
Okay, so I'm not sure I totally understand your question, but you're asking about how...
Do they use things like potassium permanganate?
Okay, so you're asking about antimicrobial compounds and if they can foster antibacterial resistance?
To reduce bacterial.
Yeah, so that's
at various stages of food preparation.
Yeah, so that's a great question. I haven't done much research in that area, but I know that there is a problem with using soaps that have antimicrobial compounds in them and then them getting into the environment and fostering bacterial resistance. Okay. Rebecca?
How did the presence of actual pathogens correlate with the different activities? And maybe more interestingly, how does that compare to your indicator data?
Yeah, that's a great question. So unfortunately, we only tested a subset of our samples for pathogens, so we didn't look at how specific activities affected pathogen levels. In those 30 samples that we looked for pathogenic E. coli, I did have 10 paired samples where we took them before and after hand washing with soap. We did find pathogenic E. coli after hand washing with soap was still present. But that says nothing about the quantity of the pathogen that was present on the hands. And then your second question, looking at the association between actual pathogens and fecal indicator bacteria. It's a good question. Again, since we have just a subset of samples here tested for pathogens, we might not have had the sample size to find associations. Did not find an association with enterovirus. We did find an association with pathogenic E. coli, but it's something that we're hoping to explore further, processing the thousands of hand and water samples that we have from the Tanzania project. Caitlin?
I was wondering if you could talk a little bit about what you think the differences might be between some of these interventions or behaviors in urban versus rural areas, because these are focused on urban areas, but you find really different things in rural areas.
Yeah, so that's a great question. Looking at the differences between urban and rural areas. So one thing that's different is that in urban areas, I think although usually water access is better, there's also just a lot more feces per square foot. So I mean, I think that something that's really true is that urban areas, infectious disease is a much bigger problem than in rural areas. But in terms of actual child mortality, it's actually a much bigger problem in rural areas because the access to medicine and healthcare is less. Is that sort of what you were getting at? Okay. Kevin?
I just had a question following up on Rebecca. So given the requirements for indicators and the prediction for pathogens and illness, do you think that those indicators, just E. coli and enterococci, are a good proxy for illness and pathogens in Tanzania or in Sub-Saharan Africa?
Yeah, so that's an excellent question. And I think it's still sort of a subject of much debate. And I mean, it's really debatable. I mean, even in here in the US, sometimes you find an association between indicators and pathogens and sometimes you don't. So I don't think they're the best indicators, but they're all we have right now. So I think that probably it would be helpful if future research could sort of work on trying to get better indicators. Jessie?
Do you think the data would have looked different if you took data from men's hands and watched men's behavior? Would that reveal anything interesting? I know women are really important in the study.
So I mean, my hypothesis would be yes. Just because moms are the ones, dealing with the kids' poop, doing the household chores, cleaning the toilet, all of those things. I mean, that being said, I think that if we all went to Tanzania and hung out in these communities for a while, we'd all have fecal contamination on our hands, just because the environment is so dirty. But I did find, when sampling boys and girls in schools in Tanzania, that girls had cleaner hands than boys.
That's true in the US.
Yeah. Okay. Any other questions? Niket?
Yeah, just a quick question on the non-water-based hand sanitizers. So do you see any cultural issues in getting that adopted, or do you see any major behavioral changes that would require and getting them adopted?
Yeah, that's a good question. And like I said, we've only done limited work on this subject, but I have been involved in a school study in Kenya where we actually provided waterless hand sanitizer to schools and then monitored hand cleaning behavior and health. And we found that students and teachers were likely to use the hand sanitizer and seemed to really appreciate it because water access in these schools was a huge challenge just because they had to go and fetch water, just like households have to. And there's usually no one responsible for going and fetching water, but if you just have hand sanitizer right there, then the kids use it. Okay, so I think, thanks so much for coming. And now we'll go to the torture chamber. Oh, my acknowledgements. Yes. Thanks for reminding me. Almost forgot. Okay. Don't want to forget the acknowledgements. So first, I have to thank my primary advisor, Jenna Davis. She's really, over the years, redefined a professor getting their hands dirty. Here she is in a toilet in Tanzania sampling poopy soil. And over the years, I've just learned so much from her. She's given me so much support. Learning how to design research, analyze data, and do field work with a sense of humor. Next, I want to thank Alexandria Boehm, my second lead advisor. All of the microbiology that I presented today would not have been possible without her. She's also been not afraid to get her hands dirty. Here she is in Tanzania, and she's also very hardcore about research, and so I've really taken inspiration from that. But here in Tanzania, in my apartment, I found her up at 3 a.m. making media for growing bacteria with a pressure cooker in my kitchen. Next, Grant Miller, my economics advisor. He's been so patient with me as a non-economist, helping me with the first part of the research that I presented today. I always walk out of his office with new ideas and a lot of work, but they're brilliant ideas, and they make my work a lot better. Next, Pavani Ram. She flew all the way here from New York as my external committee member. She's my hand-washing guru. I really have looked up with her. I look up to her a lot. She's done amazing field work in low-income countries looking at hand-washing behavior, and I'm really thankful that she came out here today. And Abby King, so nice of her to come and take time out of her busy schedule at the medical school. She's given us actually a lot of insight on our work in Tanzania on how to look at health behavior theory and how that plays a role in our work. So I really have to first and foremost thank Tim Julian, because the third part of the research that I presented today, he was really he co-designed the research, and he did most of the lab work. And here he is. For some reason, all the pictures I have are people sampling toilets. So here he is sampling a toilet. Sarah Marks, she's my office mate. She's also come to every field project I've ever done. Somehow she shows up in Tanzania and helps me with my field work. I don't know how she does it. And Vala, my other office mate, I wanted to mention her too. She's been such a source of encouragement and support. And all these other people, I don't have time to name all of them. They've helped with my research in some way. Couldn't have been done without them. And just in general, the POOP group and the BAME group and my E-IPER cohort, and also the administrators of E-IPER and CEE. And finally, friends and family, my parents, Jesse and Zach are in the audience today, my housemates. And last but not least, Ian, my boyfriend. He's been so patient when I spend months and months away in Africa doing field work. Once he said that Africa was my first love and that he was my second, but it's not true. That's it.